ASD is associated with profound metabolic disruptions involving central nervous system. Among the hallmarks of the ASD, according to Kern and Geer publication in the journal Frontiers of Neuroscience, is low-grade inflammation in the brain that is manifested by microglial and astrocytic activation, pro-inflammatory profile of cytokines, and nuclear factor kappa light chain enhancer of activated B cells, NFKB activation. Moreover, activated microglia and astrocytes, pro-inflammatory cytokines, and aberrant NFKB activity can ultimately create an environment of excessive brain inflammation that leads to destruction of critical brain tissue and structures. ASD is associated with profound metabolic disruptions involving central nervous system. Brain has very high energy demands, consuming approximately 20% of energy while accounting for only 2% of total body weight, and demanding a great amount of adenosine triphosphate, the ATP, to maintain ionic gradients essential for neurotransmission and plasticity. In addition to that, mitochondria are involved in the proliferation, differentiation, and maturation of the neural stem cells, formation of dendritic processes, developmental and synaptic plasticity, and cell survival and death. The prevalence of mitochondrial disease in the ASD population is estimated to be about 5%, which is 500 times higher than that found in general population, when in general population it's only 0.01%. The prevalence of abnormal metabolic biomarkers is even higher, suggesting that as many as 30% of children with ASD may experience various metabolic abnormalities. Almost 1/3 of autistic children have documented elevations in plasma lactate and the lactate to pyruvate ratio, and the levels of many other mitochondrial biomarkers such as the pyruvate, carnitine, and ubiquinone are significantly different between ASD and the controls. 60% of ASD individuals who have seizures have biochemically confirmed mitochondrial disease. One of the crucial pathogenesis pathways of ASD is an early inflammatory process, including maternal-fetal immune interactions and resultant immunological dysfunctions occurring in ASD individuals. The prevailing hypothesis is that some combination of immune factors, including maternally developed antibodies to fetal brain, prime microglia in such a way as to impede its normal functions of directing neuronal migration. Previously, it was established that enzyme N-acetylgalactaminidase has increased levels in the blood of children with ASD. N-acetylgalactaminidase, also known as nagalase, is an enzyme that catalyzes the process of deglycosylation of the GC protein that, in its turn, inactivates its conversion into the regulatory protein macrophage activating factor. GCMF is an immunologically important protein responsible for macrophage activation. Thus, nagalase diminishes the body's macrophage activating capacity. It was also demonstrated that the cannabinoid receptor type 2 (CBD2R) signaling was significantly downregulated in peripheral blood mononuclear cells extracted from autistic children, suggesting that endocannabinoid system dysregulation could be involved in ASD-mediated immune impairments. It is well established that immunotherapy with GCMF forte reduces negalase levels in ASD patients that correlates with reduced severity symptoms and manifestations of ASD. The possible mechanism of action of immunotherapy in ASD can be two: stimulation of matured macrophages M2 formation plus immunomodulation via the vitamin D link transcription factors and via the CB2R receptor system. Excitotoxicity. It's a phenomenon pertinent to both adult neurodegenerative disorders and ASD. It is a pathological neuronal excitation through overstimulation of neurons by excitatory amino acid receptors of glutamate and aspartate, the so-called NMDA receptors. As a result of that, there is an increased influx of calcium in the postsynaptic membrane, overproduction of free radicals, increased oxidative stress in the neurons, mitochondrial dysfunction in the neurons, accumulation of pro-inflammatory molecules, neuronal swelling, dysfunction, and finally apoptosis, the death of neurons. Same mechanisms in ASD, same mechanisms in ALS, Alzheimer's disease, and many other neurodegenerative disorders.